Thank you, everyone. I'd like to start with a story. The story of a husband and wife team of microbiologists who went through something truly unimaginable. Steph had been married to Tom for 17 years before he fell ill. For Thanksgiving that year, they decided to travel, and not to some rural town to visit their grandparents, but to the pyramids of Giza. Being microbiologists, they had heard that Giza was a hotspot for a certain deadly bacteria called Acinetobacter baumannii. Being microbiologists, they weren't too concerned about this because they had been exposed to so many dangerous parasites and viruses in the past, and they figured that if one of them did contract the bacteria, an easy round of antibiotics would wipe it out in no time. Boy, were they wrong. Tom would end up in a coma, septic, and dying. With antibiotics being of no help, Tom surely would have died. Late one night, Steph was researching alternate medical solutions to Tom's case, and she discovered something interesting. She discovered something called the clinical application of bacteriophage. She found that bacteriophage are only 30 nanometers across, and yet they are the most prolific mass murderers on the planet. Bacteriophage is a virus that specifically hunts and kills bacteria, just like how the Ebola virus or coronavirus hunts and kills animal cells. Bacteriophage, there are approximately 10 to the 31 bacteriophages on this planet, which is 10 times the number of bacterial cells. Bacteriophage is made up of 6 distinct parts. The capsid head, which holds the DNA, the collar, which holds the top and the bottom parts of the bacteriophage together, the sheath, which is a needle-like mechanism, the tail fibers, which are those little legs that you see on the screen, and the spikes, which secure the bacteriophage onto the bacteria. Bacteriophage were first discovered in 1915 by a French-Canadian microbiologist who found that something was killing his bacterial samples. So he decided to filter these samples, and what he found amazed him. He found that on the other side of the filter, nothing was showing up. This meant that whatever was killing his bacterial samples was the same size or smaller than the bacteria itself. He immediately reasoned that it must have been a virus, and he would spend the next 10 years of his scientific career defending his theory that the thing that was killing off his samples was a virus and not an enzyme like many of his colleagues thought it was. Now, the way in which bacteriophage latches onto bacteria is very interesting. We call bacteriophage a highly selective organism because it has certain protein complexes on its tail fibers, which is the thing that attaches to the bacteria, that match specifically with certain proteins that are only on specific bacterial cells. So if a scientist wants to only kill one type of bacterial cell and not kill a wide range of bacterial cells, some good, some benign, like an antibiotic would, they can use their sample of bacteriophage to target that specific bacteria. Now, when a bacteriophage lands on the surface of a bacteria, an irreversible process is started. It injects its sheath, which is that needle-like structure I was talking before, into the bacterial cell, injecting its DNA into the bacteria. Once the DNA is into the bacteria, it takes over the organelles of the cell, forcing those organelles to produce more phage. Once the amount of phage becomes so abundant inside of the cell and the pressure becomes so immense, the bacteria pops, killing the bacteria and sending the phage flying in every direction, their sole purpose to hunt and kill more harmful bacteria. Now, when Steph discovered this, she had hope for the first time in a while that she would be able to save her husband. So she reached out to phage labs all over the country. The only response that she got was from a professor at Texas A&M University, and he said that if she could send a culture of Tom's bacteria, that he could check if any of his phage samples would have been a match. Fortunately, the scientists at A&M struck gold. And less than a week later, scientists at A&M and doctors at Tom's hospital were flooding his body with a virus in a last-minute attempt to try to save the man from the grasps of death. Within a couple of weeks, he recovered, infection-free. This was an amazing clinical example of bacteriophage working in a clinical setting, and it proved to scientists that bacteriophage could be used to help millions of people a year fighting terrible bacterial infections. Now, you might be wondering, why is drug-resistant bacteria such an important issue? Well, first let's think about how bacteria becomes drug-resistant. When you have a group of bacteria and you introduce an antibiotic, not every single one of those bacteria are going to die. You're going to have a couple left over who had a mutation to avert that drug. And you might be thinking, oh, well, you just have a couple left over, maybe your immune system, your body will be able to wipe that out. Well, the problem is the way in which bacterial cells divide through a process called binary fission, where one cell turns into 2, 2 into 4, 4 into 8, 8 into 16, etc., makes it very difficult to control this growth. In theory, one drug-resistant bacterial cell can produce 16 million offspring in less than 8 hours. If we don't mitigate this problem, it's estimated that drug-resistant bacteria will cause more deaths than cancer by the year 2050. Now, what's so amazing about bacteriophage is that when the bacterial cell wall starts to mutate to avert these drugs in phages, the phage, because it's an organic organism, will try to mutate itself to keep up with the mutation of the bacterial cell, which is something that antibiotics just simply can't do. If we wanted to keep up with this rapid mutation and growth of these new drug-resistant bacteria, we would have to be making a new class of bacteria every 3 years. Before a couple of months ago, we hadn't created a new class of bacteria, or a new class of antibiotic since 1984. Now, you also might be wondering why, what does drug-resistant bacteria or bacteria in general do to the human body? Well, when you introduce a bacteria or a drug-resistant bacteria into the bloodstream, it causes something called septic shock. And your arteries and your vasculature starts to crush together and close up in on itself. It also causes pooling of blood in your peripheral tissue. What this means is that not enough blood is going to get to your vital internal organs. And when a human goes into organ failure, it's basically never good. Now, fortunately, there is hope. Bacteriophage research centers are becoming more and more prevalent all around the country. The hospital that treated Tom, which was the University of San Diego, created the Center for Phage Therapeutics. Likewise, the Yale School of Medicine just treated a man back in 2018. He came in with an aortic aneurysm, and they replaced his aortic arch to treat that aneurysm. And post-procedure, the surgeons noticed clear signs of a dangerous bacterial infection. Now, the surgeons treated this infection with multiple rounds of end-stage antibiotics, but he was still quickly deteriorating. So they tried to get end-stage palliative care approval from the FDA to conduct a clinical study using phages on this guy. And the FDA came back to them and said, sure, you can do this because he's probably going to die anyway. So they introduced the phage using IV in the first ever IV introduction of phage into a human. And within weeks, he was better. This was another incredible clinical example of how bacteriophage could perform at a higher level than an antibiotic. Recently, there was a study conducted where there was a big group of people who all had the same bacterial infection. Half of these people received a phage treatment, and half of them received a antibiotic treatment. 87% of the people who received the phage treatment recovered fully, whereas only 64% of the people who received the antibiotic treatment fully recovered. This is another example of phage not only doing the job of an antibiotic, but outperforming it in a clinical setting. Now, it's really important to understand that a lot of the work that we've been doing is not yet complete. We don't have solutions to issues like how do we keep bacteriophage stored at a close to freezing temperature when many countries on Earth can't afford to do so? How are we going to mitigate the risk of toxic shock when introducing a bacteriophage onto a gram-negative bacteria can cause it to release toxins into the bloodstream? These are all issues that we have to address, and as a society, we have to push for more funding and more research and development onto the bacteriophage field. I had a deep connection with my grandfather because of our mutual love for science. He would send me articles ranging from anything from black holes to dinosaur bones. And when he started a bacteriophage company after leaving his career in law, I was inspired. Shortly before his passing, he got a bunch of FDA approvals for different phage products, and he was ecstatic. He said that he was excited not only for the future of the company, but for my future, for our future, a future that was hopefully just a little bit safer. Thank you.